Hello students, we are learning biology, and in biology in last few lectures we have studied sexual reproduction in plants, vegetative reproduction in higher plants, and in last lecture we have studied the sexual reproduction. In that, the typical flower, parts of typical flower, calyx, corolla, androecium and gynoecium. So these two are essential walls, androecium and gynoecium. And if you want to know the structure of androecium has different stamens. So all stamens together in a flower is called androecium. And if you want to know the structure of single stamen has anther. इसमें क्या होता है? Anther होता है, and filament and anther are connected through a connective. So this is the anther and this is the filament. So entirely this is called a stamen. So anther is very important part in sexual reproduction in plants. Anthers are of two types. Anthers may be a monothecous anther or maybe a dithecous anther. Monothecous anther means anther has a single lobe containing two pollen sacs. जब anther में एक lobe होता है, and in that lobe if the anther has two pollen sacs or two microsporingia, then that anther is called monothecous anther. Mono means single. The second one is dithecous anther. The anther consists of lobes and each lobe contains two microsporingia. So dithecous anther has two lobes and each lobe has two microsporingia. So total there are four microsporingia. So dithecous anther is then called Tetrasporongiate anther. So that we have seen last time in last lecture. Okay. Now what is the structure of Tetrasporongiate anther? When you take a transverse section of anther, the anther shows structure that is called epidermis and inner layers. So basically anther has epidermis. ये outer layer जो दिखाई देता है इसे epidermis, और उसके अंदर जो होता है just beneath the epidermis you can see the hypodermis. And some of the hypodermal cell, these are developing or getting separated into primary parietal cell and sporogenous cell. ये जो hypodermal cells होते हैं anther में, they get differentiated into two different things, outer primary parietal cell और दूसरा inner sporogenous cells. So this outer parietal cell later on again differentiate into anther wall. ये किस में develop होता है? Anther wall में. So anther wall is originating from the primary parietal cells, or the parietal cells उससे क्या होता है? तैयार anther wall, and the sporogenous tissue forms the microspore mother cells, and the microspore mother cell later on gives rise to pollen grains, or microspore. So this is what differentiation of cells from immature anther to mature anther. So suppose when you take a section, transverse section, you can see the structure like this, and then if you enlarge it or see under microscope, so what you can see in the transverse section of anther, इसमें क्या दिखा देता है आपको? इसमें दिखा देता है connective tissue, बीच में ये connective tissue आपको दिखा देता है. This exactly the in the center on either side you can see the two lobes. Okay, so this is one of the lobe of anther and this is another lobe. So a tetrasporongiate anther, a dithecous anther has two lobes. ये वाला एक lobe और ये वाला एक lobe. Correct? So this is one lobe और ये दूसरा वाला lobe है. Correct? So this is what tetrasporongiate anther, and there are two pollen sacs. This is, or single one, this is another one. So these two pollen sacs, or these two pollen sacs. So total there are four pollen sacs. So if you want to know the detailed structure of this is the connective, which is essential for giving nourishment to the anther, and then the outermost wall of this anther is called epidermis. What is epidermis? Epidermis is outermost wall of the anther. Just inside that, or just beneath the epidermis, second layer is what? Endothecium. दूसरा क्या है? Endothecium layer है. Okay. So outer is epidermis, second one is endothecium, and just inside the endothecium you can see the middle layers. ये जो middle layers है ये endothecium के अंदर it may be can say one or two middle layers वहाँ पर होते हैं. These are what middle layers, and then the innermost layer of the anther wall is tapetum. Tapetum क्या है? एक innermost layer है. So this is the tapetum, and this is innermost layer of the anther wall. And inside the tapetum, you can see sporogenous tissue or microspore mother cell. Pollen mother cells or microspore mother cell, these are present just you can say inside the tapetum. Okay, so in the Dithecous Anther, if you see the structure, in the structure what you can see, there are four can say sporangia or pollen sac, and that's why it is called Tetrasporongiate Anther, or can say anther with four pollen sac. Outermost layer of anther is called epidermis. Just inside the epidermis you will get endothecium, that's the second layer. So first layer is epidermis, second layer is endothecium, third is what? Middle layer. ये के दो सेल थिक होता है मिडल लेयर. सो दिस इज ऑन नंबर 3. नंबर 4 पे आ जाता है tapetum. This is the innermost layer of the anther wall, and inside the tapetum नंबर 6 पे आपको दिखाई देता है sporogenous tissue. Sporogenous tissue are microspore mother cells which are developed from the sporogenous tissue. Microspore mother cells are you can say pollen mother cell. So this is the structure of a typical anther. एक anther Dithecous anther होता है, उसका structure है. Okay. Now at the maturity or once the anther become mature, the walls of anther get open. Dithecous anther की wall open हो जाते हैं, and the pollen grains which are developed inside they'll come out, and then they take part can say in pollination, or through can say different agent they are transferred from anther to the stigma, and then fertilization takes place. Okay. Now if you see the layers of anther, the epidermis is the outermost layer. ये सबसे बाहर वाला number 1 is epidermis. ये epidermis है. What is the function of epidermis? The epidermis is what? Protective in function. It gives protection to the inner cells, or can say microspore cell को ये protection देता है और inner tissue को protection देता है. Then there are what? Second one, the second layer ये क्या है? Endothecium है. Endothecium and middle layer, and ये जो दो लेयर होते हैं या ये के दो लेयर होते हैं middle layers and endothecium along with can say epidermis takes part in the essence of anther wall. Anther wall का dehiscence कब होता है? At the maturity of can say anther, or can say after development of pollen grains, the anther dehisces, and ये epidermis, endothecium and middle layer 3 they take can say they have a role in dehiscence of anther and can say liberation of pollen grain. Tapetum is most important. ये tapetum है सबसे important है, that gives nourishment to microspore mother cell. इसका main function क्या है? Give nourishment, or can say to nourish and nurture, or can say to give material, food material to microspore mother cell for further development. ये nourishment का काम करता है. And the microspore mother cells होते हैं, so these are forming microspores. So meiosis takes place in microspore mother cell, and then microspores are formed. Okay, so these are the different layers. Epidermis number 2 पे होगा endothecium, endothecium. नंबर 3 पे रहेगा middle layers, middle layers maybe 1 or 2 cell thick, and just उसके नीचे यानी कि उसके अंदर middle layers के अंदर ही यानी innermost layer of anther wall is tapetum, tapetum, and within the tapetum उसके अंदर यानी कि inside the tapetum, not within, inside the tapetum you will get sporogenous tissue. उसके अंदर क्या रहेगा? Sporogenous tissue. And this will later on give rise to microspore mother cells. they form microspore mother cells, and this microspore mother cell divides with can say meiotic cell division and forms microspores. ये क्या तैयार कर देते हैं? माइक्रोस्पोर तैयार कर देते हैं. Microspores, correct? So this is what the structure of anther when you can when you take transverse section and observe under microscope. So this is what the part of anther है. anther का एक part है, एक can say pollen sac है और एक can say sporangium है. In that you can see microspore mother cells. ये क्या है? Microspore mother cells. So these are what? The microspore mother cells. If you take a single microspore mother cell, एक microspore mother cell से 4 pollen grains तैयार होते हैं. Okay? So this is single microspore mother cell. This microspore mother cell undergoes meiotic cell division. उसमें क्या होता है? Meiosis होता है. Meiotic cell division means reductional cell division, and from a single microspore mother cell, you can see the formation of 4 microspore. Initially, it is in the form of tetrad. So this is called microspore tetrad. So there are 4 can say microspores. They are connected to each other. After that, or can say after maturity, they get separated, and then from that you can see the formation of 4 microspores. The single microspore, if you see the structure of single में होता है nucleus. ये nucleus है, and this is microspore wall. Correct? So once this microspore become mature, that forms a pollen grain, and pollen grain has wall. The outermost wall of pollen grain is called exine. The inner wall is called intine. Somewhere on pollen wall you can see there is very thin can say structure. Only can say inner wall is can say thin. Outer wall is not that much can say developed there. So that areas are most essential areas from where the pollen will come out. So that is called germ pore. So pollen grain has outer wall exine, inner wall intine, and the germ pores, and inside that you can see the nucleus. So microspore mother cells are diploid one, and then once they undergo meiosis, they form can say tetrad, and a single microspore, if you see, it is what? Having haploid number of chromosomes. So this is can say initial stage, initial development. Microspore mother cell divides meiotically and forms microspore, and at the maturity the microspore develops into a pollen grain, a mature pollen grain. Now we will see the structure of pollen. Pollen grain का structure कैसे होता है ये अब अभी देखेंगे हम लोग. So if you see the structure of pollen grain, in pollen grain the outermost wall of the pollen grain is called exine. जो outermost wall होता है pollen grain का उसको क्या बोलते हैं हम लोग? Exine बोलते हैं. And this exine is made up of sporopollenin. किससे बनता है? Sporopollenin. So sporopollenin is a structure, उससे बनता है ये exine, और ये exine ऐसा tough होता है, it is not degraded by any enzyme. किसी भी एंजाइम से डिग्रेड नहीं होता. सो क्यों नहीं होता? बिकॉज इट इज मेड अप ऑफ व्हाट? दिस स्पोरोपोलिन. इसको डिग्रेड करने वाला एंजाइम अभी तक मिला नहीं है. द सेकेंड वॉल, दैट इज व्हाट? इनर वॉल ऑफ एंड दैट पर्टिकुलर पोलन ग्रेन इज कॉल्ड इंटाइन. आउटर वॉल पोलन ग्रेन की होती है एक्साइन से बनी हुई, जिसका एक्साइन नाम है स्पोरोपोलिन. इनर वॉल इज मेड अप ऑफ सेलुलोज एंड पेक्टिन, उसको बोलते हैं हम इंटाइन. And inside that intine you'll get a cytoplasm. ये पूरा क्या है? साइटोप्लाज्म है. And then some areas are there, ऐसे कुछ areas वहाँ पर ये outer inner wall एकदम thin दिखाई देते. Means outer wall दिखाई देते नहीं, only inner wall you can see. So thin inner wall ही दिखाई देते. So these areas are for thin areas in the wall of pollen grain. So from where pollen tube will come out. क्या वहाँ से क्या आता है? pollen tube बाहर आती है. So pollen tube बाहर आती है. क्यों आती है? Because वहाँ पर उसको जगह है बाहर आने के लिए. So that area is called germ pore. इसको हम क्या बोलते हैं? germ pore बोलते हैं. So this is the structure of pollen grain. इसके अंदर एक न्यूक्लियस है, करेक्ट. एक न्यूक्लियस, अराउंड उसके साइटोप्लाज्म है, पूरा एक साइटोप्लाज्म, जर्म पोर, बाहर जो जगह देता है पोलन ट्यूब को आने के लिए, जर्म पोर, देन इंटाइन इनर वॉल ऑफ द कैन से पोलन ग्रेन एंड आउटर वॉल. सो हम एक बार फिर से देखते हैं, रिवाइज करते. एक्साइन इज मेड अप ऑफ स्पोरोपोलेनिन, एक टफ वॉल होती है. इनर वॉल इंटाइन मेड अप ऑफ सेलुलोज एंड पेक्टिन. ये दोनों को मिला के हम क्या बोलते हैं, स्पोरोडर्म. इसको बोलते हैं स्पोरोडर्म. सो वॉल ऑफ पोलन ग्रेन इज कॉल्ड स्पोरोडर्म, इसको बोलते हैं स्पोरोडर्म. इनसाइड दैट विल गेट साइटोप्लाज्म एंड न्यूक्लियस. क्या मिलता है, साइटोप्लाज्म एंड न्यूक्लियस. और जहाँ पर थिन एरियाज है उसको हम बोलते हैं जर्म पोर. दिस इज द स्ट्रक्चर ऑफ एंथर. इसके ऊपर कभी कभी क्वेश्चन आ जाते हैं एग्जाम में. सो if you want to describe the structure of pollen grain, simply you can say the outer wall of pollen grain is exine, inner wall is intine, then it has a cytoplasm, nucleus and germ pore. So this is the structure of pollen grain. Now this pollen grain is most important structure in angiosperm, because from this pollen grain a male gametophyte is developed. मेल गैमेटोफाइट इससे बनता है, किससे बनता है, पोलन ग्रेन से बोलता है. नाउ पोलन ग्रेन इज अ स्टार्टिंग पॉइंट फॉर डेवलपमेंट ऑफ मेल गैमेटोफाइट और फॉर्मेशन ऑफ मेल गैमेटोफाइट. मेल गैमेटोफाइट फॉर्म करने के लिए, तैयार करने के लिए शुरुआत कहाँ से होती है, पोलन ग्रेन से होती है. सो दिस इज द पोलन ग्रेन. जैसे हम लोगों ने देखा, आउटर वॉल इज एक्साइन, इनर वॉल इज इंटाइन, इसका ये साइटोप्लाज्म है. दिस इज द न्यूक्लियस ऑफ पोलन ग्रेन. नाउ दिस न्यूक्लियस ऑफ पोलन ग्रेन डिवाइड्स माइटोटिकली. इसमें क्या होता है, माइटोटिक सेल डिवीजन होता है और इक्वेशनल सेल डिवीजन हम उसको बोलते हैं. एंड दिस न्यूक्लियस, करेक्ट, ये जो न्यूक्लियस होती है ये डिवाइड हो जाती है एंड देयर इज फॉर्मेशन ऑफ 2 सेल्स. इससे क्या बोलते हैं, 2 सेल बनता है. सो बिकॉज ऑफ माइटोसिस, ये माइटोसिस पोलन ग्रेन में होता है. पहली बार ये माइटोसिस होगा और ये माइटोसिस होके क्या बनेंगे, 2 सेल बनेंगे. सो 2 सेल्स आर फॉर्म, वन इज वेजिटेटिव सेल और दूसरा जेनरेटिव सेल. पहला सेल जो लार्ज सेल होता है, उसका इरेगुलर न्यूक्लियस, लार्ज सेल विथ फूड न्यूट्रिएंट एंड ऑल, ये बड़ा वाला सेल उसको बोलते हैं वेजिटेटिव सेल. और एक छोटा एक सेल तैयार होगा, दिस इज जेनरेटिव सेल. नाउ दिस जेनरेटिव सेल, नाउ क्या होगा, मे बी आफ्टर सम कैन से टाइम इट एंटर्स इन द साइटोप्लाज्म ऑफ वेजिटेटिव सेल. उसमें फ्लोट हो जाती है, अंदर चली जाती है, फ्री हो जाती है. करेक्ट, क्या होता है, फ्री होता है या उसके अंदर चला जाता है. दिस व्हाट इट्स फ्लोटिंग or can say enters in the cytoplasm of vegetative cell. So now this is what the pollen grain. You can see 2 cells in this. One that is vegetative cell and its nucleus. दूसरा the generative cell and its nucleus. बराबर, ये 2 cells आपको pollen grain पे दिखाई देते हैं. Okay, so in initial stage, first stage में क्या होगा, pollen grain का जो nucleus है, it will divide and form 2 cells, vegetative cell and generative cell. Okay, now this generative cell will enter in the cytoplasm of vegetative cell, उसमें free हो जाएगी and then it will can say again take part in development of male gametophyte. पहली बार माइटोसिस हुआ था, माइटोसिस हो गया. ओके, नाउ ये जेनरेटिव सेल में अभी क्या होगा, सेकेंड माइटोसिस होगा एंड इट विल टेक प्लेस ओनली इन दिस जेनरेटिव सेल. ये जेनरेटिव सेल है, इसमें माइटोसिस होगा दूसरी बार. सेकेंड टाइम माइटोसिस होगा, किसमें होगा, जेनरेटिव. सो ओनली जेनरेटिव सेल डिवाइड एंड आफ्टर डिवीजन इट फॉर्म्स 2 मेल गैमेट. माइटोसिस होगा, इक्वेशनल सेल डिवीजन होगा और तैयार क्या होगा, 2 मेल गैमेट तैयार होंगे. नाउ दिस 2 मेल गैमेट एंड अ वेजिटेटिव न्यूक्लियस ऑफ वेजिटेटिव सेल, ये आपको मेल गैमेट दिखाई देता है और ये वेजिटेटिव न्यूक्लियस दिखाई देती है. दिस इज द स्टेज, हम इसको क्या बोलते हैं, दिस इज द स्टेज At this stage, most of the pollen grains, they get, can say, separated or come out of the anther and then take part in fertilization. क्या करते हैं वो? Anther dies at this stage and the pollen grains along with this, you can say that, you can say this is male gametophyte with two male gametes. Now इसको हम क्या बोलेंगे? Male gametophyte बोलेंगे. At this stage they'll come out from the anther, or maybe in some other cases, कुछ ऐसे भी cases होंगे, maybe after this male gamete will enter in the pollen tube तैयार होगा, उसमें जाएंगे. At this stage also in some of the cases they'll come out from this. Okay. Now what we have seen? We have seen the second mitosis from generative cell, generative nuclear divide हो जाती है, and then there is a formation of two male gamete. Okay. Now at this stage anther से क्या होगे? pollen grain बाहर आ जाएंगे. बाहर आके क्या होगा? Now at this stage this pollen grain with two male gametes, correct? ये जो दो male gamete है, हम उसको ग्रीन कलर से अभी दिखा देते हैं, two male gametes. Okay. And ये जो vegetative nucleus है, at this stage pollen grain will come out from the anther and get deposited on the stigma. जो स्टिग्मा रहेगा कार्पल का पार्ट, और कैन से हम उसको स्टिग्मा बोलते हैं, ये स्टिग्मा पे ये डिपॉजिट हो जाएगा. सो सपोज दिस इज द स्टिग्मा, स्टिग्मा पे ये डिपॉजिट हो गया. सो वंस इट गेट डिपॉजिटेड ऑन स्टिग्मा, स्टिग्मा पे डिपॉजिट होने के बाद द जर्म पोर जो होता है, ये वाला जो जर्म पोर है, इसी एरिया से from this germ pore you can say what will happen? The inner wall intine will, can say, come out and form a pollen tube. ये क्या होगा? Pollen tube. So at this stage once it get deposited on the stigma and get sufficient, can say, stigmoid fluid, जो fluid इसको मिल जाता है, एक liquid जिसमें pollen sticky इसमें से because of that fluid sugary material होता है, वहाँ पर pollen tube grow हो जाती है. This pollen tube will, can say, grow or will come out, intine से बनेगी पूरी pollen tube आपको यहाँ पर, and this the male gamete will get transferred in a pollen tube, and then this pollen tube will reach to the female gametophyte for fertilization. Now who will direct this? किसको ये direction का काम दिया गया है? This tube nucleus, or you can say nucleus of vegetative cell, that, can say, takes initiative, forms, can say, pollen tube and directs the male gamete in the pollen tube, and then male gamete will enter in pollen tube and then later on take part in fertilization. So we will see the steps again. Pollen grain is the starting stage of male gametophyte development, and formation of pollen tube, entry of pollen tube, can say, in a style through stigma, and, can say, moment of, or can say, transfer of these two male gametes along with a tube nucleus in a pollen tube. That is what the latter part of male gametophyte development. So these are the different stages of male gametophyte development, and this is the male gametophyte, a small plant where male gametes are produced, and these male gametes will take part in fertilization in angiosperm. So this is what a typical, say, male gametophyte development in angiosperm, a part of sexual reproduction. Okay. So how pollen grains are produced and how, can say, male gametophytes are developed from pollen grain and male gametes are formed. So these are what all the steps that we have studied. Okay. So this is what important, can say, part of the syllabus, and maybe, can say, you'll get many objective questions in, can say, CET, or maybe, can say, MST CET, या नीट में भी क्वेश्चन आ सकते हैं, या महाराष्ट्र बोर्ड के एग्जाम में भी आ सकते हैं, और मे बी कैन से small can say questions, short questions भी इस पे आ सकते हैं. So this is what important part, development of male gametophyte. So what we have seen in today's lecture, we have seen the structure of anther and development of male gametophyte. Okay, so definitely आपको इसका फ़ायदा होगा. So learn from this video. इसको एक बार बारीकाई से स्टडी करो. It will help you to can say get to know how to draw the diagram, can say transverse section of anther, and how can say draw the diagram of can say development of male gametophyte, different stages of male gametophyte and all. Thank you very much.